All right, so it's an interface, and we got the membrane, the nuclear membrane, and the centrioles and the chromatin, which is the DNA, and so in interface, the chromatin, the DNA doubles, and so do the centrioles, and then we move into prophase one, and in prophase one, the nuclear membrane dissolves, and the chromatin thickens and turns into chromosomes. And we have four sets of chromosomes, or four chromosomes, which are two chromatids. And the homologous pairs find each other, creating a tetrad. And the last step of prophase one is the centrioles travel to the poles and create spindle fibers. And then in metaphase one, the spindle fibers pull the in prophase one, so the DNA crosses over and the chromosomes cross over, and we're going to represent that by exchanging the pen caps. And then so in metaphase one, it pulls the two homologous pairs, the tetrads, onto the equatorial plate, like by the spindle fibers. And in anaphase one, the spindle fibers pull apart the chromosomes, and then in telophase one, a nuclear membrane forms around the chromosomes on each side of the cell, and the cleavage furrow starts. And we're going to pretend that instead of splitting, so it would normally split to two cells before prophase 2, but instead of doing that, we're just going to have a string that goes across like this. Then in prophase 2, the nuclear membrane on each side dissolves, and the centrioles get pulled to the poles of each side of their own cells, and create spindle fibers. And in metaphase 2, the chromosomes get pulled to the equatorial plate, and in telophase 2, the chromosomes get pulled apart, and they're just their own chromatids, which are also chromosomes because they have centromeres in each one, and the chromosomes get pulled apart in anaphase, and in telophase 2, then nuclear membranes form around each of these cells, and we're going to just pretend we did that for all four of them, and a cleavage furrow starts, and cytokinesis happens, and the cell divides again. So you start with one cell, and it goes through prophase, metaphase, anaphase, telophase 1 and 2, and it becomes four cells before going back to interphase. And in each cell, you now have a different chromosomes, because there's variety through the crossing over, because none of them are the same. And also the chromosomes turn back into chromatid at this stage. So you have four new cells that all have different chromosomes and chromatin in them, which are now chromatin in them. And that is meiosis,